Dear students, Assalamu alaikum. Welcome again to our program. If we recall our last program, we were discussing about the types of cell division, and we discussed in detail mitosis, the cell division in somatic cell for the purpose of growth, for the purpose of repair. Today we will be going with another variety of cell division, which is requirement for the organisms going through the sexual reproduction. Because the organisms are going to produce gametes in the form of sperm and ova, and the beauty of mitosis was in production of identical offspring. And here, if we are going to keep the same number of chromosomes, sperm having 46 fusing with the ova having 46, so that will be 92 sperms in the zygote, in the new offspring, and that will be a new species, not the human being. So there is a need of a variety of cell division in which the number of chromosomes are reduced to half, so that once the fertilization is going to occur, so the chromosome number that is maintained to a constant level. So that is called as a reduction division, and the terminology we are using, that is meiosis. The word meo means to reduce, a reduction division in which the number of chromosomes are reduced to half. So it is going to occur only in the production of gametes. Gametes mean sperm and ova with reference to animal, and spores with reference to plant. So going to go with the certain important features about the meiosis and the number of chromosome, going with the few slides. So meiosis for the formation of gametes, that is the ova and sperms. And if we are going to look at the life cycle, so the adult individuals, they are going to produce the male and female gamete, and they are going to fuse at the time of fertilization to give us the zygote. So here requirement is to reduce the number of chromosome to half. So once fertilization is going to occur, they are maintained to the full level. And from the zygote onward, from a single cell, whole of the individual is going to develop by the process of mitosis, which is going to maintain the same number of chromosome in the adult body. There are some facts about mitosis, that as in mitosis, so in case of meiosis also, the interphase is there before the start of this division, in which the chromosomes are going to replicate. The cells undergoing the process of meiosis, they are going to go through the two nuclear division. Two meiotic divisions are there, meiosis I and meiosis II. So also called as reduction division. Original cell is diploid, and instead of 2, 4 daughter cells are produced that are haploid, having half the number of chromosomes. Daughter cells contain half the number of chromosomes as the original cell, and it is used only in the production of gametes. So that is testes in male, spermatogenesis, and oogenesis, that is the formation of ova in the ovaries of female. Starting with the 46 double-stranded chromosome, after first meiotic division, 23 double-stranded chromosome they are there. After second meiotic division, 23 single-stranded chromosomes are there, and it is going to occur only in the germ cells. Why do we need meiosis? It is the fundamental basis of sexual reproduction. The two haploid gametes are brought together through fertilization to form a zygote that is diploid. So these are the sperms fusing with the ova to maintain the same number of chromosomes. So this is the sperm having 1 and the 3 chromosomes, ova having the 3 chromosomes. So they are going to fuse to give us 2 and diploid, the 6 number of chromosomes in the living organism at the time of fertilization. So before that, the DNA must replicate, because that have to divide. So replication is the process of duplication of the chromosome. This was a single chromosome, so that have to duplicate into 2 sister chromatids held together at the centromere. So meiosis is going to occur in the 2 parts. first meiotic division, so in which the homologous chromosomes are going to associate and then they are going to separate, and second meiotic division in which the chromatids are going to separate from each other. So these are the homologous chromosomes which are going to separate in the first meiotic division, and sister chromatids are going to separate in the second meiotic division from the diploid cells. So ultimately these are the 4 haploid daughter cells which are produced by this division. So in the first meiotic division, again the same phases are there, that is the prophase 1, metaphase 1, anaphase 1, telophase 1. So prophase is further classified into early prophase and the late prophase, in which the chromosomes are going to appear and the certain events are going to occur, and then the arrangement of the homologous pairs in the middle, that is the metaphase 1, separation of homologous chromosome, and then the formation of the 2 daughter cell as a result of first meiotic division. In the prophase 1, so the first event is condensation of chromatin to take the form of chromosome, and homologous chromosomes, they are going to appear, crossing over is going to occur, while in the late prophase, so condensation of chromosome is complete, spindle fibers are going to form, nuclear envelope begins to break. Prophase 1 is further classified into leptotene, zygotene, pachytene, diplotin and diakinesis. The prophase 1 of meiosis is a very lengthy and highly specialized phase. This is the main difference between the mitosis and meiosis, prophase 1. So what is there in the prophase 1? In the first substage of prophase 1, leptotene, the chromatin, the genetic material which was there in the condensed form, so that is going to coil and recoil to take the form of chromosome, and the homologous chromosomes are going to appear. And I think you must be familiar with what the homologous chromosomes are there. The 2 chromosomes which are similar in size, in shape, in position of centromere, having the same gene, controlling the same trait, these are called as homologous chromosome. They are having some difference also. They can be having different allele of the same gene, and they are different in their origin. One is coming from our father and other is coming from our mother. So these are going to associate in leptotene. Then in zygotene, so the homologous chromosomes are going to pair lengthwise to form the bivalents, and this is the process of synapsis. As a result of synapsis, tetrads are produced. Then in pachytene, what is going to happen? Crossing over, there is exchange of genetic material from the chromatid, one chromatid to the other chromatid. So this exchange of genetic material is called as crossing over. There is reshuffling of the genetic material to give us variety in the offspring. So crossing over in pachytene, in diplotene, so the chiasmata, they start breaking away, there is a repulsion which is going to start, and in diakinesis, so there is disappearance of the nucleolus, nuclear membrane, spindle fibers get attached, and all other events of prophase, they are being completed. Going to continue with some of the slides. So what the tetrads are? So this is one chromosome, this is the homologous part of the same chromosome, and what is going to happen that these 2 homologous chromosomes, they are going to combine together in the form of tetrads, and this process of pairing of homologous chromosome is called as synapsis, crossing over. So basically these were the homologous chromosomes. The chromatids of these chromosomes are going to cross. At this point of cross is called as chiasmata. At this point of cross or chiasmata, there is exchange of genetic material. So this chromosome have taken the genetic material of that one, and it is the vice versa. So this genetic recombination, exchange of genetic material, that is called as process of crossing over, which is going to occur in the prophase 1. Going to look on another slide of crossing over. So these were the two homologous chromosomes. This is the tetrad which is produced by the process of synapses, and now they have exchanged these materials. So these blue parts, so they are given to this brown one and vice versa. Looking at the animation of crossing over, so these are the two homologous pair, and they are going to join together like in this format. If we are going to have a closer look, so the part of one chromosome is going to detach and it is going to attach with the other chromosome. So this is the process called as crossing over. Going with the metaphase 1, in the metaphase 1, the homologous chromosomes, so they are going to align along the equator of the cell. In case of mitosis metaphase, so these were the single layer of chromosome, and here it is the double layer of chromosome, homologous pairs, they are being arranged. In anaphase 1, what is going to happen? The pull is exerted by the spindle fibers, and these are the homologous chromosomes which are going to be pulled apart. These are the full chromosome which are attached at the centromere. Sister chromatin remains attached at their centromere. And then in the telophase 1, again nuclear envelope that is going to reassemble, spindle fibers are going to disappear, and cytokinesis divide the cell into two. As a result of first meiotic division, so each of the daughter cell is going to receive the half the chromosome, that is called as reduction division, and now the sister chromatids carry identical genetic information. Meiosis II produce gametes with one copy of each of the chromosome and thus one copy of each of the gene. So looking at the prophase 1, so these were the two daughter cells which were produced. Again the nuclear membrane is going to break, the spindle fibers they are going to reform, and in metaphase 1, so these chromosomes now they are single. So they are going to arrange at the equator or the midline of the cell, and usually the alignment is at the right angle to the first division. So these chromosomes they are going to be aligned in the middle in the metaphase 2. Anaphase 2, so again sister chromatid they are separating and they are pulled apart to the opposite pole from the equator, and these are the poles which are being shown in this. Going with the telophase, so in the telophase again the reappearance of the nuclear membrane, decondensation of chromosomes, spindles are going to disappear, and cytokinesis divide the cell into two. Results of meiosis are the four daughter cells which are being produced. So these are haploid cell with one each of having one copy of the chromosomes. One allele of each gene is present in all of these. Different combination of allele for different genes along with the chromosomes are there, and this is due to the crossing over. As a result of meiosis, four daughter cells are produced, and these daughter cells are different from each other. The cause of difference is the two factors. One is the crossing over, that is the exchange of genetic material during prophase 1. The second cause is the random alignment of chromosome during metaphase 1. So any chromosome, the pair of homologous chromosome, any member can be on the right or left side. So this random distribution of chromosome at metaphase 1, that is also going to give us the genetic variety. In case of mitosis, the two daughter cells which were produced, they were identical, but here the four daughter cells which are produced, so they are different from each other due to crossing over and due to random assortment of chromosome at metaphase 1. Looking at some of the animations of process of meiosis, so this is one of the cell having these 6 chromosomes in different. So these chromosomes, first they are going to replicate their chromatids. So they are replicating their chromatids. Then these are going to arrange in the middle in the form of homologous pairs. Crossing over is going to occur, exchange of genetic material, and then they are going to move towards opposite pole, half of the homolog on one side, half on the other, and a cell membrane is produced. So two daughter cells are produced as a result of first meiotic division. Then the second meiotic division is just like the mitotic division, in which the chromatids are going to split apart and 4 daughter cells are produced. Each daughter cells are going to have the different arrangement due to crossing over and random assortment of chromosomes. Looking at another animation of meiosis. So this is one of the cell under the prophase 1, appearance of the homologous chromosomes, and then these homologous chromosomes are going to pair by the process of synapses, and then these homologous chromosomes, they are going to arrange at the equator or in the midline of the cell. By the movement of these spindle fibers, they are going to attach at these chromosomes, and the site of attachment is the centromere of the different chromosome, while this was the centromere of the same chromosome in case of mitosis. Here, so this spindle fiber is attaching on this centromere, and lower one is going to attach to the centromere of the second chromosome. Pull is exerted by the spindle fibers, and then these full chromosome made up of 2 chromatids, so they are pulled apart during first meiotic division or meiosis 1. This is the anaphase. Then the telophase is going to occur, that is the 2 daughter cells are produced after that, after the first meiotic division, and number of chromosomes are reduced to half. So this first meiotic division is called as the reduction division, and now it is going to undergo with the second meiotic division. Again there is disappearance of the nuclear membrane, the chromosomes are going to align in the middle, and this time, so these are the chromatids which are going to split apart. So 23 chromatid moving on one side, 23 moving on other side, followed by the telophase, as a result of which the 4 daughter cells, they are being produced, and then it is the uncoiling of these chromosomes to take the form of chromatin. Comparing the process of meiosis and mitosis in this animation. So first, looking at meiosis, the summary of the difference between the 2 processes. So here association of the homologous chromosomes are there in the first meiotic division, as a result of which, so the crossing over is going to occur. No such process is going to occur in case of mitosis, and then again the splitting of the cell into 2, and again these cells, so they are going with the second meiotic division with the splitting of cell into 4. So these are the 4 daughter cells. In case of mitosis, so this is a single division of nucleus and single division of chromosome, and only 2 daughter cells are produced. So these daughter cells, they are 100% same, they are identical, there is no genetic reshuffling, and here the genetic reshuffling is there. So you can look at this part. So this is these are the genes from other chromosome which are taken by this gamete, and these are the genes which are taken from some other chromosomes. Going with the process of gametogenesis, that is oogenesis in female and spermatogenesis in female, formation of male and female gametes. We have gone with the process of meiosis. Now this meiosis is going to occur only in the formation of germ cell, and these are gametes. The gametes are produced where? In the gonads, and the gonad in male is called as testis, and gonad in female is called as ovary. So humans, they are going to have 2 testes. They are there in a sac called as scrotum. Within the testes, so there are many septa are there, and there are a lot of seminiferous tubules are there, which are the factory for production of sperm. At the lining of these tubules, so there are the cells which are called as spermatogonium, and this spermatogonium undergo the first meiotic division, second meiotic division to give us the sperms. Similarly, in the ovary of human female, so there are cells which are called as oogonium, and these oogoniums are also going to go with the first meiotic division and second meiotic division to give us the mature ova in the ovary. Looking at these details of these processes, spermatogenesis occurs in testes. 2 division produce 4 spermatids. Spermatids mature into sperms, and a man on an average 250 million sperm is being produced per day. So many of sperms are produced per day in human males. This is the spermatogonium, the diploid cell, the mother cell, which is going to give us the haploid cells. So it is undergoing the first meiotic division, as a result of which you can say the two daughter cells are produced, and then it is going to go under second meiotic division to give us 4 spermatids, which are going to mature in the form of sperms. Going with the oogenesis occurs in the ovary. It is slightly different from the spermatogenesis, that the two divisions, although they produce 4 cells, but the 3 of cells, they are small in size, called as polar bodies, and they are going to die away. Only one egg is being produced. Polar bodies, so they are going to die because of unequal division of the cytoplasm. Immature eggs are called as oocytes. Starting at puberty, one oocyte mature into a ovum, and every 28 days, that is during one menstrual cycle, one ova is released. So basically this is the oogonium, the basic diploid cell that is going to differentiate into primary oocyte undergoing the first meiotic division, and here there is 2 daughter cells are produced. So having the equal genetic material, but they are having unequal cytoplasm. This is a small cell having a little cytoplasm. This is given the name of polar body. So this secondary oocyte is also undergoing the process of second meiotic division, giving one big cell having a lot of cytoplasm and other small cell having a very little cytoplasm. So this is called as polar body. So these 3 polar bodies, they are going to die away, and only one polar body that remains functional. So number of division 1 and 2, 1 in mitosis, 2 in meiosis, daughter cells 2 in mitosis, 4 in meiosis, genetically identical cell in case of mitosis and genetically different cell in case of meiosis, and chromosomes, so same as the parent, but here number of chromosomes are reduced to half. Mitosis is occurring in somatic cell, meiosis is occurring only in the sperm cells. Mitosis is going to occur throughout the life, while the meiosis is going to occur at the sexual maturity, and the role of mitosis is in growth and repair, while the role of meiosis is in sexual reproduction. Dear students, we have compared the process of mitosis and meiosis. So one division is called as a general division. All the divisions which are going to occur in our body, they are mitotic division. Only germ cells, they are the exception. So they are having this special division of meiosis. And the 4 daughter cells which are produced as a result of meiosis in comparison to 2. So these 4 cells are different from each other. This is giving us the diversity. Why we are different from our father and mother, and why the brothers and sisters, they are different from each other. This is the genetic variety, variation. There is a tendency of offspring to be similar to their parents, which is called as heredity. So some of the genes we are going to inherit from our father, and some of the gene we are going to inherit from our mother. But we are not the exact copy of father and mother, because this is the genetic variations which are given, and the causes of genetic variation is number 1, that is crossing over. Number 2, this is the random assortment of chromosome. Number 3, it is the random fusion of sperm and ova. Millions of sperms are released during a single ejaculation, and only one is required. So any sperm among these millions, it can fuse with the ova. So it is a new genetic recombination. Then it is the random mating of individuals. So there are, you can say, the random marriages are there. Any person can have the option to marry 1, 2 or other persons. So this is the random mating of individual, random fusion of gamete, crossing over and random assortment, that is going to give us the genetic variety. And this genetic variety, this is very, very important, because environment is constantly changing, and those individuals who are going to fit into to survive in a specific environment, so they are going to live. So basically this genetic variety which is given by the process of meiosis, that is the basis of evolution. So looking at the last slide of our presentation about the value of variation. So the variations, these are the differences between the members of population, and this is the result of meiosis. If there is no meiosis, so all of us, we are almost the same clone of our parents. So we are not the clone of our parents, we are different from our parent and from each other. And this causes diverse population that over time can be stronger for survival in a specific environment. Dear students, we have completed the topic of continuity of life. How the life is going to continue either by the process of mitosis, that is from a single cell, whole of our body is going to develop, that is mitosis, maintaining the stability of gene, genetic stability. And second was in sexually reproducing organism, a reduction division, so that at the time of fertilization, the number of chromosomes remains the same, but the genetic variety is given. So that is going to give us genetic diversity in comparison to genetic stability to mitosis. Inshallah, in our next program, we will be looking at the abnormalities occurring in mitosis and meiosis. So these are the chemical accidents and mishaps which are going to occur leading to various diseases. Till that time, take care and Allah Hafiz.